Hi everyone, in today's lesson we're going to be looking at the second half of protein synthesis, and that is the translation of the mRNA code. For this lesson, you need to be able to discuss protein synthesis and explain the role of ribosomes, codons, anticodons, amino acids, and peptide bonds. Now, using that, you should also be able to interpret an amino acid chart, also known as a degenerate code. These are keywords associated with today's lesson, very similar to what's in the learning objectives. It's really important that you have a good understanding of this terminology and are able to apply it. So, before we start looking at translation, we're going to just have a summary of what we've learned so far. Now, I want you to try and use these diagrams to help you explain what we have talked about. So here we've got a double helix structure and a deoxyribonucleic acid nucleotide. Down here we have the single helix, so obviously this is DNA and this is RNA, and because this is an RNA nucleotide, it has a different color pentagon just to represent that this is ribose sugar and this is deoxyribose sugar. So you should be able to connect those two things and explain what the link is. Moving along from that, I would like you to look at this diagram here and explain, write down, discuss with someone else what is happening and what stage of protein synthesis is taking place. What molecule is being formed, and can you explain the difference between the coding strand and the template strand? Now, this diagram has been enlarged at the bottom here so you can see the bases, and I'd like you to write a sentence, if you haven't already done so, about the differences between the coding strand and the template strand and the base sequence on the RNA and the DNA. Okay, so hopefully you have done that and you're confident with your knowledge on transcription. Now we're going to be looking at translation. So in translation, remember, we have left the nucleus, so our mRNA coding sequence has left the nucleus. We're now in a cytoplasm and attached to ribosomes. Now before we go any further, I need to remind you that translation is the process of forming a protein using tRNA polymerase. So that's an enzyme, because it ends in A's, that helps to synthesize this reaction that you see in this diagram. So the codons on mRNA, so remember a codon is a group of 3 bases, will bind with their complementary anticodon, which is a complementary group of 3 bases on tRNA. As that happens, as these tRNA molecules bind to mRNA at their complementary base sequences, it brings with it an amino acid. So you can see this tRNA molecule, which has the anticodon sequence AAG, brings the amino acid phenylalanine, or PHE as it says here. And we see that the complementary mRNA sequence is UUC. So what will happen is this tRNA molecule will bind at the complementary base sites to the mRNA molecule, and it brings with it an amino acid. Now we have mentioned that this process all occurs in the cytoplasm of the cell at ribosomes, and it's the ribosomes that actually read this mRNA code and helps the tRNA molecule to bind to it, as I've just described. Now, you'll notice here, we've got in the diagram, it says a growing polypeptide chain. So these single amino acids join together, and the bond that forms here is a peptide bond. Now, in order to form a peptide bond, we require ATP because this is an energy-consuming process. Now, here we are looking at the amino acid table, also known as the degenerate code. Now, the genetic code uses the principle that there are 4 different bases, which are adenine, thymine, cytosine, and guanine if we're looking at DNA. So this makes up triplets. So we know that DNA comes in triplet codes, which make up 64 different combinations. So this is more than what we need. So 4 to the power of 3 is 64. We only have 20 amino acids, so it gives us lots of scope for different combinations. The degenerate code, which you're seeing in this table, is simply a code with several different meanings. Okay, so if we look here at the amino acid proline, we have several coding sequences that all equal proline. And similarly with leucine, and valine, and alanine, and so on. Now, there are some exceptions to this rule. So we have methionine, as you see here, AUG. There's only one of that, and that is a start sequence, and you also have a stop sequence. So for example, up here you have UGA, which is our stop sequence. Now, some of the genetic code is universal in all organisms, and this can be really handy when we start looking at genetic engineering and gene transfer. You'll learn more about that in year 12. So here I'm just showing you another version of the degenerate code or amino acid table with the full name of the amino acids underneath. Now, you do not need to ever remember this code or remember the names of the amino acids. Using the 3-letter code is usually good enough, and if you are asked to name the amino acid, you'll be given a table. The skill here is being able to look up what the amino acid is. So for example, if the first position of base was C, we would then look to the second one and the third one. So the second position is at the top, so let's say that the second position was also C. That would bring us into here. And if we said that the third one was A, we look across at A and we see that the code is CCA, and therefore the amino acid code is pro. And if we go down to our table, we can see that pro is proline. So this is a skill that you need to practice, and you need to understand that this is what we are looking for. And sometimes you'll be asked to work backwards from this using your knowledge of base pairing. Okay, so now we're going to talk a little bit about ribosomes. The ribosomes, as we said before, are the units where the mRNA code is read. Now, ribosomes occur in 2 subunits. So the whole thing is a ribosome, and we have the larger subunit on the top and the smaller one at the bottom. And it creates this groove here in the middle. And that is actually really significant, because what it does is it allows the mRNA code to be fed through the middle and read. So the assembly of amino acids in the correct sequence only occurs because the ribosome allows this mRNA code to be read, and it allows the interaction of mRNA and tRNA, and therefore bringing the correct amino acids. So now we're going to be looking a little bit at tRNA molecules. This diagram is from your textbook, and we're just going to take a moment to study it carefully. So we can see at the bottom of the tRNA molecule there is a group of 3 bases, and this is the anticodon sequence. Now, the anticodon sequence, as we've mentioned before, is complementary to a specific amino acid sequence. We've got hydrogen bonds forming between bases here, and this is what allows the bases to contort and conform, and this is what gives tRNA its distinctive shape. Now, at the top, this is a really important part, apart from the anticodon sequence at the bottom. What we have here is an amino acid that is being carried by this specific tRNA molecule. So because it has the code CGG, it only carries the amino acid alanine. They will not carry any others. So they are specific. So here, just to summarize, we have the second type of RNA that we talked about in protein synthesis, and this is transfer RNA. Now, transfer RNA is also made in the nucleus, but it's found in the cytoplasm. It has a set of triplet of bases in our RNA called anticodons. The anticodons are complementary to the mRNA codons, and remember, it's always a group of 3 bases. So at the top of the molecule, as we said, there is an area of 3 unpaired bases where the amino acid will join. Now, the amino acid at the top corresponds to the anticodon sequence at the bottom. So now that we understand a little bit about the structure of tRNA and how it corresponds with mRNA, I'm just going to explain how translation occurs. So here, this pink line represents our mRNA code, and the blue blob in the background is the ribosome. So we can see that our tRNA molecule is sitting on top of the complementary codon sequence in mRNA. Now, what we can see here is the mRNA code is AUG, and the corresponding amino acid is methionine, which is actually the start sequence. So this section of code on mRNA initiates that we are starting to translate this code into a functional protein. Now, I haven't shown it in this diagram, but what you should see, or you should know, is that if this is the mRNA coding sequence, the tRNA anticodon should be complementary. Therefore, it will be UAC. As we can see, the next tRNA starts to come in carrying its specific amino acid, and it will join up at the next codon sequence, which is GCC. Therefore, the tRNA coding sequence is CGG, and this brings the specific amino acid alanine. As we move along, we can see that the tRNA molecules are now sitting adjacent to each other in the ribosome, and the amino acids are also sitting adjacent to each other. And what starts to happen is a peptide bond forms. So once that bond has formed, the tRNA molecule will release the amino acid and move away, and it will go back into the cytoplasm to collect another amino acid. Now, because the tRNA molecule has a specific anticodon sequence, it can only ever pick up a corresponding amino acid. So for example, this tRNA molecule has the anticodon sequence of UAC, so therefore it can only ever pick up methionine. It cannot ever pick up alanine. So what we can see here is that empty tRNA molecule has gone away, as I just described, and the next amino acid, which we saw in the picture above, has come along to the ribosome, and it's gone through the same process that we talked about before, and the tRNA molecule that was sitting in the first position has moved to the second position here, and everything gets shifted along. So the ribosome runs along the mRNA code and brings in those tRNA molecules, and they are read. And as that code is read, the tRNA molecules bring their corresponding amino acids, and the base sequence, the amino acid sequence continues to grow. Okay, so that's quite a lot of information that we've discussed so far. So what I would like you to do is to pause the video at this point and see if you can discuss, so just using words, discuss what we've talked about and try to use all of the key terminology. Okay, hopefully you were able to do that. Here I've got a little hyperlink to a video on YouTube that very briefly covers translation. It's quite useful. I'd recommend that you watch that. But I'm just going to go through the stages of translation. So the first thing that happens is mRNA leaves the nucleus via the nuclear pore. It moves to the cytoplasm where it will attach to a ribosome. And remember, ribosomes can be free in the cytoplasm or they can be attached to the endoplasmic reticulum. The ribosomes will start to read the mRNA code. Remember, we have the large and small subunits on the ribosome, and the mRNA goes through that groove in the middle. Transfer RNA, or tRNA, carries amino acids to the ribosome, and usually the first amino acid in any polypeptide chain is methionine, and this is known as the start codon. tRNA anticodons will complementary base pair with the mRNA coding sequence, hence the codon for methionine is AUG, whereas in tRNA the anticodon is UAC. After the start codon, a second tRNA molecule attaches itself to the next codon on mRNA in the same way, and the two amino acids that are attached to the tRNA molecules will start to form peptide bonds. Now, because this is a bond formation process, it uses ATP. Once that bond is formed, the tRNA molecule moves away, leaving the amino acid behind. The third molecule binds to the next codon on the mRNA, and its amino acid will then bind to the first two and then move away. Now, this process keeps happening, and it will continue as the mRNA is read through the ribosome until there is a stop codon that has been reached. Now, once that's happened, the polypeptide chain will move away from the ribosome because translation has been completed. It will, of course, move to the other organelles to help package and process that newly formed polypeptide so that it can become a functional protein. So here I've just got a diagram, a little cartoon explaining what we've talked about. You might want to use this to help write down some notes. You don't need to worry about the E, P and A sites, that's a bit more advanced, but what I do want you to focus on is the mRNA moving through, the complementary anticodon and codon sequences, and the growing polypeptide chain. So again, another little summary just to show you how translation occurs. So this is our mRNA leaving the nucleus via the nuclear pore. A ribosome in the cytoplasm attaches to the mRNA sequence. And we know that each group of 3 bases in mRNA is known as a codon. The corresponding tRNA molecule will move into place and it will attach to the codon using its anticodon. And when the second one comes along, as we've mentioned, a polypeptide starts to grow and we get a peptide bond forming between adjacent amino acids. Now, once that peptide bond has formed, the empty amino acid will move away to the cytoplasm to collect another amino acid, and another tRNA molecule comes in and takes its place. Now, this keeps happening along the length of the mRNA code as the ribosome reads it. Eventually, we get to a stop sequence, and the polypeptide knows to detach. Now, this will keep happening many times. So here what I've shown you is that there's actually many ribosomes that will perhaps read that same piece of code. So you won't just in reality want one protein, you'll actually want thousands of them. Now, what's happening in the cytoplasm, just to let you know, is that we have got a free amino acid that wants to attach onto its tRNA molecule so that it can go back and protein synthesize again, and this process also consumes energy. Okay, so just a little summary of what we've learned in today's lesson. We know that translation is the second stage of protein synthesis, whereby amino acids assemble to make proteins. We know that this happens as a result of the DNA sequence, which was copied from DNA by mRNA. The genetic code for this copied sequence comes from the DNA in the nucleus. The mRNA makes a copy of the DNA code, which is translated into a sequence of amino acids which are attached to tRNA molecules. It's the ribosomes which are the site of protein synthesis, and as I've mentioned, they can be free in the nucleus or they can be attached to the endoplasmic reticulum. So that's the end of this teaching lesson. I would like you to make sure that you have completed pages 9 to 11 of your booklet. Please make sure that you also complete the questions here from your chapter review in your textbook found on pages 246 and 247. At the end of this PowerPoint, there's also additional questions and exam practice for you to go through.